Good evening, everyone. So this week we will shift a bit from melanogenesis and melanocytic disorders. Previously, we have discussed the process of pigment formation and followed by pathomechanism of vitiligo, followed by oculocutaneous albinism. Now, not this week onwards, we'll see how this video is received, and maybe we will continue this video and delve more into dermatopathology. But I thought that it would be a good idea to discuss dermatopathology, and what good way to discuss is first to know what is the normal histology of the epidermis. So first, if you know how the epidermis looks like under a microscope, you will be easily able to find out what is wrong with the epidermis when you're analyzing the slides from different disorders. And you'll be easily able to find out how a cell is deviating from its normal maturation. As we know, epidermis is concerned is consisting of majorly the keratinocytes, and how a keratinocyte transforms as it moves from the basal layer and gradually shifts towards the corneum, the stratum corneum, and how it is actually shed off. And any problems in that process, in that procedure or that process, actually leads to the different appearances of corneocytes. And when these different appearances we see in a disorder, along with a lot of different features we see at derm-epidermal junction and the dermis, we take all the findings together in a constellation. Okay, we aggregate all those findings that we see in a biopsy, and then we correlate that with the clinical history provided and the clinical picture. And if you are the one seeing the biopsy, you are in a better position to actually analyze what a disorder is trying to tell you, and you are able to, number one, confirm the diagnosis. Number two, you can take out any kind of doubts that you had in clinically while examining the patient. So, first of all, any biopsy that you analyze, especially in the initial stages in residency, while you're going to understand more about dermatopathology, the description starts from the epidermis and we go downwards to involve the dermis and the subcutis. So if you're describing a slide, it's better to describe it going from top to bottom. And with time, when the experiences increases, you might go directly to the most evident and the most prominent findings. But it should be, even if you're not describing it as such from top to bottom, the idea to examine the slide should be from top to bottom. That is, you start from the topmost layer of the epidermis and you go downwards till you reach the end of the section or the subcutis. Okay. And then you are able to, when you have made a note of each and every findings that you see, you take into account the presentation of the entire slide, and that will help you into analyzing the slide in a much better fashion. So slowly, and in a group of different kind of video lectures, we'll try to understand the basics of dermatopathology, and gradually we'll move towards the individual skin disorders and the histopath pictures. So without much further ado, we'll understand how a normal epidermis looks under a microscope, and we'll start our video lecture on histology of the epidermis. So epidermis is embryologically ectodermal in origin, and the name means layer upon dermis. Epidermis, it's above the dermis. It does not have any blood supply, and the nutrition is through diffusion. While see this, if this is the section and this is the dermoepidermal junction, the epidermis does not have a blood supply, and all the nutrients diffuses in between the cells or inside the cells from the dermis onwards. Remember, the dermal vessels are somewhat in the mid-dermis. They can reach up to the papillary dermis, but most of the time it's in the junction of reticular and papillary dermis. And that is where the nutrients kind of diffuse to reach the epidermis. The thinnest area for the epidermis are the eyelids, 0.5 millimeters the normal average thickness, while the thickest areas are the palms at 0.8 mm and soles, which is 1.4 mm. And that we know that normal epidermis is thickest at the soles. The cells of the epidermis are stratified squamous epithelium. Stratified squamous means the cells are arranged in a layer. If this is again the section, different layers are formed by the cells. So this stratification or strata formation. Strata means layer. So it is stratified squamous epithelium. And this we are talking about the corneocytes. Okay. Corneocytes or keratinocytes. Now remember, what's the difference between keratinocyte and a corneocyte? Keratinocytes are all the cells of the epidermis, which will form the skin layer. Okay. So all these skin layer forming cells the cells of the stratum corneum, granulosum, spinosum, basal. While corneocytes are the term used for the cells which are in the upper layers, stratum corneum, where the cell has undergone such a change in morphology that they are now flattened and ready to be shed off. And then they are known as corneocytes. Okay. Other cells that are there in the epidermis are the dendritic cells, the melanocytes, Langerhans cells, and Merkel cells. Now, melanocytes are responsible for the melanin in the skin. Langerhans cells are antigen-presenting cells, and Merkel cells are neuromodulatory or neurosensory organs. Neurosensory organs. So we will discuss in detail only the keratinocytes and the appearance, maybe a bit about melanocytes, but the Langerhans cells and Merkel cells appearance are not part of this video lecture. We'll just focus our discussion on keratinocytes. Let's move forward. So let's start from the description of each and individual epidermal layers, and we'll describe them from basal to corneal. Okay. Now I know it goes against what I said in the first slide that we should always describe from top to bottom, but right now we're just learning. So when you describe a slide, you go from top to bottom. But as learning is concerned, we'll start from the most recognizable layer or most easily visualized layer because it is at the junction of the dermis and the epidermis, and that layer is stratum basale or the basal layer. It is also known as stratum germinativum. It is also known as stratum malpighi or the Malpighian layer. It consists of a single layer. So remember basal layers. The stratum basale is a single cell layer, only one cell layer thick. The cells are columnar. Sorry, cells are columnar to cuboidal, and there's a basal oval nuclei with prominent nucleoli. Okay. They are attached to basal membrane zone with hemidesmosomes, and we have discussed hemidesmosomes in detail in previous lectures. If you want a more theoretical lecture, you can just go back and it's in the playlist. Now, the cells of stratum basal continuously divide, and that is why they are known as mother cells of the epidermis, that because they give rise to all the cells of the epidermis. So what actually happens is that if you have a basal cell like this here, a basal cell, gradually it will turn into a spinosum layer, and then a granular layer, and then a corneocyte. Okay, and since all the keratinocytes are arising from the stratum basale, the cells of stratum basale are known as mother cells of the epidermis. The predominant keratins are keratin 5 and keratin 14, and these are there in the basal layer only. As the cell moves from basal layer to the superficial layer, the keratin gradually shifts to keratin 1 and 10, but these are not the only keratin, but predominantly keratins change from 514 to 110 as the cell moves up. Let's move forward. So in this photomicrograph, this histopath picture, you can see all the layers of the epidermis. You can see a very thickened stratum corneum, a little refractile or a little blurry layer that you can see is stratum lucidum, very thin layer. And if you can see this blue colored basophilic layer, this is stratum granulosum. If you see the layers in between, which has a little bit of white dot in between, that is stratum spinosum. And just at the junction of dermoepidermal junction, you have stratum basale. Okay, so remember stratum basale is one layer thick, so only one cell layer is known as stratum basale. Rest all are predominantly made up of stratum spinosum and a little bit of stratum granulosum. And most of the corneocytes are residing here at the level of stratum corneum. Okay, mention in the comments if you can recognize what is this structure, this star-shaped structure. If you know what it is, mention it in the comments. I'll tell what it is in the next week. So this gives you a more clearer picture because the thickness of stratum corneum is less. It's more of a thin skin biopsy. And here you can see again the stratum corneum. This diffractile pink color layer is stratum lucidum. Stratum corneum is stratum lucidum. While a bit of a bit of basophilic bluish color is stratum granulosum. In between is stratum spinosum, and this brown pigmented single cell layer thick is stratum basal. So just go through from top to bottom, evaluate analyzing all the individual layers of the cell, layer of the epidermis. Let's move forward. So this is the diagram that I had previously shown while discussing melanogenesis, and this is just a schematic representation of a basal cell. So what actually I'm trying to show here is that the basal cell are columnar. Okay, they are columnar. It has a prominent pigmented nuclei, and it has a melanosome cap, a melanosome cap. So if in the basal layer you are seeing these kind of cells, let's see this cell. Let me just point it out. This cell, okay. So if you look at this cell has brownish pigmentation in between, and this is not a melanocyte because melanocytes don't have pigment. They transfer their pigment what they have made to basal cells, and basal cells collect those pigment in their cytoplasm. So all the brown colored cells that you can see in the basal layer are actually basal keratinocytes. Okay, so the melanocytes are actually not pigmented. So for example, here you can see a melanocyte. Here you can see a melanocyte. You can see there's no cytoplasmic pigmentation present. In fact, here you can see a melanocyte. There's no cytoplasmic pigmentation. Let me just change the color. Yeah, here you can see a melanocyte. Here you can see a melanocyte because there's no pigment in the cytoplasm. I mean, here you can see a melanocyte, and you can see this kind of vacuole, this empty space. So that is one marker of a melanocyte. But remember, the cells that you see in the basal layer, which are pigmented, are actually basal keratinocytes. They are not melanocytes. Let's move forward. So the second layer that we'll discuss is stratum spinosum. It's also known as prickle cell layer because of the thorns or a spinous appearance. It's prickly, so prickle cell layer. But remember, this is not a natural appearance. It's the appearance after processing. So what actually happens is, in a normal individual, you have keratinocytes. Okay? So you have keratinocytes, and they are attached to their surrounding cells by desmosomes. Okay, we have discussed this in the basement membrane class. So when you are processing the tissue sample, the slice from the tissue sample in the lab, the cells shrink. And when they shrink, they're only attached to their neighbors via desmosomes. So only the part which is attached to desmosomes does not shrink. And that not shrinking of the part looks a bit spinous, like the cells have spine. And that is why the name was given stratum spinosum, but that is just an artifactual appearance after processing of the tissue sample. Okay. This cell layer is 5 to 7 layers thick of nucleated polygonal cells, and the keratin now is 1 and 10. Remember, it is 514 in the basal layer, gradually shift to keratin 1 and 10. So let's move forward. So in this picture, you can see the spinous layer very easily. There's also an additional pathological finding of spongiosis, but let's just focus on the spines that you can see. So let me change the color again. Can you see this as the keratinocyte, and these are the spinous processes? These are all the areas where the cell is attached to its neighbor cells, which has not retracted because of desmosomes, giving it a spinous appearance. That is why stratum spinosum. Okay, let's move forward. Now the third layer from bottom up is stratum granulosum. It is a 2-3 cell layer of flattened spindle cells. So as we move from a columnar basal cell, and then a spinous cell, and now you have a bit of flattened spindly cell. Okay, spindle-like cell, like a myocyte, and then you will have a flattened corneal site. Okay, so that is how the cell moves from basal upwards. So at the level of stratum granulosum, you have a flattened spindle cells, and that cell layer is 2 to 3 layer thick. Now the granules, and that is why the name comes from the granules, stratum granulosum. The granules are dense, basophilic, keratohyaline granules. Remember basophilic. Basophilic means they will appear blue on H&E stain. And they contain majorly lipids. And these granules are known as odd-line bodies. And also the cells of stratum granulosum has proflagellum. And these two entities, outline bodies, profilagrin, along with a lot of other entities are important in the process of keratinization. So what actually is happening is that the cell, as it is moving from basal layer to the upper layers, it is converting itself to a corneocyte, a keratinized corneocyte. And that is why the granules are now rich in the products of keratinization or the raw materials of keratinization, and you have increased outline bodies in profilagrin. And that is what is happening. So in the granular layer, you can see them distinctively because now the granules are very large. And because of increased lipid content, it acts as a barrier to fluid loss. It prevents the loss of moisture. The nucleus is pycnotic, that means it is condensed and darkly stained, and the organelles begin to disintegrate. Remember, in corneocytes, the organelles are not present, they are disintegrated. You have a dead cell, somewhat of a quasi dead cell, okay? The keratin filament bundles are more compact, irregular, and dense, but you don't see them in H&E. And they have lamellar round granules, and these are known as keratinosomes. But you won't see these structures on H&E. You won't. You will appreciate the granular nature, the basophilic granules in the cells of stratum granulosum. But other structures would require a higher magnification, special stains, electron microscopy, and all that. So right now we'll restrict ourselves to H&E. Let's move on. The fourth layer from bottom up is stratum lucidum. The stratum lucidum is translucent layer of flat, dead, clear, non-nucleated cells. So remember, in stratum lucidum, the cells have lost their nucleus. Okay, so they are flat, dead, clear, non-nucleated cells. Stratum lucidum is thick on palmaroplantar skin. Now remember, wherever the skin will be thicker, the thickness of its individual layer will also be more. And that is why the stratum lucidum is actually in normal skin biopsy a very, very, very thin layer. But when the tissue itself is very thick, it makes the stratum lucidum easily visible. Okay, it stains stronger and more optically refractile, making it more lucid, more clear. Okay, and that is why it is known as stratum lucidum, a clear layer, more optically refractile layer. It consists of compact keratin filaments. Okay, let's move on. So look at this photomicrograph. You can easily appreciate the single cell layer basal keratinocytes. You can see the spinous process of stratum spinosum. Okay. You can see the basophilic granular layer of stratum granulosum. And above that, you see this kind of pink thin layer. Let me change the color of the pen. This pink thin layer here, and that is what is known as stratum lucidum. Okay, so from now onwards, if you are looking at a biopsy slide, try to find this layer, stratum lucidum, in between the stratum granulosum and stratum corneum. Let's move forward. Stratum corneum is the last layer now. So it is the outermost dead layer consisting of flattened non-nucleated cells, which are known as squams. Squams means flattened cell basically, and these are what we call as corneocytes or cells of the stratum corneum. The thickness of the layer varies with region, and the outer layer is shed continuously by the process of just shedding of the skin. So the natural shedding of the skin, it is shed continuously. The cell membrane is thick due to involucrin on the inside and a lipid layer on the outside. So involucrin is another protein involved in keratinization, and now the inner side of the cell membrane is lined by a layer of involucrin. In this cell, the profilagrin, remember we read about profilagrin in stratum granulosum, has now been converted to filagrin. And filagrin is a very important molecule required for keratinization. Maybe in the future when we'll discuss the process of keratinization, I'll tell you what exactly filagrin does. Let's move forward. So here you can see you have a flattened corneocyte and the red, and it is surrounded by yellow part. The yellow part is the lipid which is outside the cell membrane, and the red part is involucrin which lines inside the cell membrane. Okay. So these bricks, these pink bricks are actually individual corneocytes. And if we attach the outermost lipid layer, you have a background of yellow which is composed of lipids. So it looks like that the brick-shaped corneocytes are sealed by a mortar or a cement of lipids, and that is known as the brick and mortar model. So it looks like that you have bricks symbolizing corneocytes, and the cement that you put in between, the mortar that you put in between, is made up of lipids, and that is known as the brick and mortar model. Okay, let's move forward. Now, melanocytes. Melanocytes, the nucleus is large, round. Okay, it is mostly over the cheek. We have discussed everything in the process of melanization or melanogenesis. There's epidermal melanin unit. That means for every 30 corneocytes, or sorry, 30 keratinocytes, they are linked to one melanocyte, through which the melanocyte transfers melanosomes to these keratinocytes. It also has a perinuclear halo. That means a bit of white space around the nucleus. So this is a schematic representation of a melanocyte. You can see the dendritic process, and these dendritic process are responsible to transfer melanin to the connected corneocytes. Melanin transmission. Or actually, to be correct, melanosome transmissions. Okay, so you can see the large nucleus. You can see this white rim, which is perinuclear halo. So that is how you understand. If the cell in the basal layer is not pigmented, it has a large basophilic nuclei, and it has a halo around it, it is a melanocyte. The pigmented cells are keratinocytes. That distinction is clear, I hope. Let's move forward. Now, Langerhans cells are also dendritic cells, the same as melanocytes, but they are responsible for antigen presentation. That means they detect antigen and present it to the circulating lymphocytes to start the immune response. One essential structure is the Birbeck granules. Birbeck granules are actually discoid, cup-shaped. So they are like a cup, like this, like cup. And when you cut them horizontally, they have this kind of tennis racket appearance. So this is a very important, very commonly asked MCQs, however, a tennis racket appearance. Okay, and that is what I'm showing in the diagram. Okay, and when they get activated, they present the antigens to the circulating lymphocytes in the lymphoid tissue and start the immune response. Now, Merkel cells are also neural crest in origin, the same as melanocytes. Remember, a few weeks back we discussed how melanocytes travel from neural crest to reach the skin. They are more commonly in the glabrous skin, that means palms and soles, fingertips, gingiva, nail bed. They are found at the level of stratum basale and the outer root sheath cell of hair. They are clear cells consisting of keratin 8, 18, 19, and 20, and they are usually seen in immunohistochemistry. You will not be able to appreciate them better on H&E. They are neuroendocrine sensory receptors and also slowly adapting mechanoreceptors. So the function of Merkel's in the skin is as a dull mechanoreceptor to find out, to sense the touch. Okay, let's move forward. So we in the past few slides we have discussed the individual layers of the epidermis, and we'll just revise them. And here you can actually see the melanocytes very clearly. Can you see this is a melanocyte? This is a melanocyte. This is a melanocyte. This is a melanocyte. This is a melanocyte. Can you see the large round basophilic nuclei, a little bit of halo around it, vacuolated appearance, looks like an empty cell at the basal layer, isn't it? All the other cells they don't have that pigment in this photograph, but all the other cells are stratum basal keratinocytes. The in-between cells, like this cells, is the cells of stratum spinosum. The basophilic granular blue cells are stratum granulosum. There you have stratum lucidum and stratum corneum. Okay. So whenever you are seeing a slide, at least for the first time or the first semester residents, try to find these individual structures. That will make it more interesting to watch the slides, and you will not miss any findings. Let's move forward. So again, you can see the stratum lucidum, stratum granulosum, stratum corneum, Stratum Spinosum, Stratum Basale. Okay. Here also you can see a melanocyte, 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 melanocyte. Okay. A group of three melanocytes hanging together. Okay. Let's move on. So now we have gone through the normal appearances of the layers of epidermis. And when we have a bit of clarity about the normal appearance of epidermal layers, we can now gradually shift our focus to pathology of the epidermis and how it looks under a microscope. So in histopathology or in dermatopathology, we have entities known as reaction patterns. So you must have heard of thoraciciform reaction pattern, vesicobulous reaction pattern, interface dermatitis, vasculopathic reaction pattern, granulomatous reaction pattern. These are all group of findings on a biopsy that points you towards a certain set of disorders or a certain set of how a skin is reacting to a pathology. So there are epidermal reaction patterns, there are dermal reaction patterns, there are mixed reaction patterns, but we'll focus our discussion on the changes in the epidermis. So all of these things that we will discuss subsequently, they are not reaction patterns per se. They are how an epidermis will react to different stimulus. And in any skin disorder, you have multiple different kind of findings, and you take all the findings together to actually strengthen your diagnosis. So with that, we'll just discuss how a different epidermis will look like under a microscope, starting with disorders like hyperkeratosis, acanthosis, hypo or hypergranulosis, spongiosis, papillomatosis, splits in the epidermis or an epidermolytic pattern, and intraepidermal abscesses or cellular collections. So gradually, we'll show photographs and discuss how an epidermis looks like under a microscope if there is these findings. So let's start with hyperkeratosis. Hyperkeratosis, hyper means increase. Let me write it in. Increase. And keratosis means keratinization. Increase keratinization. Clear? So this is hyperkeratosis. It means increased thickness of stratum corneum, more than one third of the total epidermis. So if you have this cross section, you have the dermoepidermal junction and you have the stratum corneum here. Let's say here. So if this stratum corneum is more than one third of the entire total epidermis, that means more than 33% of the entire epidermis has only stratum corneum, you can say it as hyperkeratosis. Okay. In hyperkeratosis or the increased thickening of the stratum corneum, two types of presentation have been described: parakeratosis and orthokeratosis. Okay. First, we'll discuss orthokeratosis. Now, ortho means straight. Okay. So, orthokeratosis means straight keratinization. That means normal, no problem in that. Okay. And it is seen in normal epidermis. Straight means straightforward keratinization, orthokeratosis. Now, as I've told you that when cell migrates from the basal layer to the upper layers, the organelles and nucleus disappear. So if keratinization is happening straightforward and normal, you will not see any nucleus in the upper layers of the stratum corneum, isn't it? In the stratum corneum, you will have no nucleus in the cells. We have told you that these are dead flattened cells. Okay. So if orthokeratosis is there, we are actually looking at a normal epidermis. Okay. Now, orthokeratosis are described into two types, basket weave and compact. So what actually happens is in compact orthokeratosis, you have a thickening of the stratum corneum, but the cells are very closely packed together. The layers are closely packed together, and this is known as compact hyperkeratosis. While in basket weave hyperkeratosis, you have empty spaces in between. Okay, empty spaces in between. It looks like a basket, the one you have in villages and all, or any kind of net type basket. It looks like a net, and that is known as basket weave orthokeratosis. But remember, in both of this type of orthokeratosis, you will not see nucleus. Okay. The second type of hyperkeratosis is parakeratosis. In parakeratosis, what happens is there is delayed maturation. So when the cell moves from the basal layer to the upper layers, as it matures, it will lose its nucleus, it will lose its organelles. And but in case of parakeratosis, that has not happened. So you will see nucleus in the upper layers. So if you see nucleus in the layers, in the cells of stratum corneum, it is parakeratosis. And parakeratosis clinically presents as scaling. So if you understand the concept that the keratinocytes reaching the stratum corneum have not matured properly, they have their nucleus and they're not able to get removed normally, they will form a scale clinically. So that is why parakeratosis means scaling. Other features like dyskeratosis have also been described, and that is because of abnormal keratinization. So in that, the keratinocytes may be misplaced. That means you have cells which are not normal looking and they will be there below stratum granulosum. So if you remember, you have the basal layer, you have the spinous layer and the granulosum layer, and then you have corneocytes. But in this keratosis, you will have abnormal corneocytes below stratum granulosum because they have not properly traveled through the process of keratinization. Now, hyperkeratosis occurs because of repeated damage to the epidermis. Epidermis skin gets repeatedly scratched. It's more of a secondary changes, and there's lichenification, there's thickening of the epidermis, and when you biopsy that area, you find hyperkeratosis because of increased proliferation of keratinocytes. Let's move forward. So these are all the indications, sorry, all the conditions. It is not an exhaustive list. There are multiple more disorders which shows hyperkeratosis. And you can easily see disorders like callus cons, keratosis plantar, chronic folliculitis, atopic dermatitis, psoriasis, all the scaly lesions, all the lesions which are thickened, all the thickened plaques, lichenified plaques, everything will show parakeratosis. Disorders like ichthyosis because of retention hyperkeratosis, you have actinic keratosis, and squamous cell carcinoma because the maturation is not proper, in basal cell carcinoma because maturation is not there, and we can see BCC can be is usually thicker. Okay, so these are all the disorders which you should keep in mind when you see hyperkeratosis on a skin biopsy. So look at this photomicrograph. Okay, I've tried to include textbook pictures so that I'm able to describe exactly what you're saying. If somehow I use different pictures, I've given the references. Here also the reference is there. It's from the Weedon's third edition, and it's a good idea that after looking at the textbook pictures, we go to our labs and we go to our OPDs and wards and look at these slides that we have available of normal slides in the sense which are usually readily available so that we can also see normal variation that occurs. So what you can do is for the next 2-3 weeks, just go and look at the epidermis only of the slide. Don't look at the dermis, don't look at any inflammation or anything else. Just look at the epidermis. And let's say you have a biopsy of dermal pathology, but you just examine the epidermis and come back home. At least do this for the next 2-3 weeks or 2-3 months till you are more acquainted in finding out the different layers, delineating the different layers of the epidermis. So coming back to this slide, you can see this is the stratum corneum from here till here. Okay, this light pinkish area or the pale pink area is the stratum corneum. And we can easily see that it is thicker than the 1/3 of the epidermis. So this is the thickness and it is thicker than the 1/3 of the epidermis. So it is hyperkeratosis. Clear? And this slide shows both type of hyperkeratosis. So if you look at, see this is the junction of stratum corneum to the stratum granulosum, and this is the other edge. So this entire is the thickness of stratum corneum. And can you see that there are no nuclei in the cells? The cells are flattened. So this is actually compact orthokeratosis. Okay. But look at this area. This area. Can you see it is filled with basophilic nuclei? And what do we call the structure when you see nucleus in the stratum corneum, we call it parakeratosis. So in the same picture, you can see orthokeratosis, you can see parakeratosis, and there are multiple disorders where you will see alternating orthokeratosis and parakeratosis. Mention in the comment section if you know at least one of these disorder, and a very characteristic disorder, very characteristic histopath finding, alternating ortho and parakeratosis. Okay, so let's move forward. Now this is acanthosis, the second reaction pattern after hyperkeratosis. Now in hyperkeratosis, what actually happens is that you have the increase in thickness of stratum corneum. In acanthosis, the entire epidermis is thickened. Okay, so there is increased thickening of the entire or the multiple layers of the epidermis. Here you can see the basket weave orthokeratosis. Okay, basket weave orthokeratosis, you know, it looks like a net. Okay, so here there's thickening of the epidermis. So if this is the skin section and this is the dermoepidermal junction, the entire epidermis looks to be thicker. Okay, and you usually do that consensus about thickening is usually made in comparison with the dermis, and it is said that if the entire epidermis is more than, let's say, 10 layers thick, so acanthosis is usually mentioned. Now acanthosis could be either irregular acanthosis or regular acanthosis. So that is one thing that usually our faculties ask, can you see regular or irregular acanthosis? So what actually happens is, if you have a cut section like this, and if this is the dermal epidermal junction, and this is the outermost edge, if the thickness, if this thickness is uniform throughout the section of the slide, throughout the biopsy section, it is regular acanthosis. Okay, but this is the dermal epidermal junction. This is the outer edge. Let me make it right. If it is thickened in some part, but thinner in some part and thickened in some part, then this is irregular acanthosis. Okay. Other thing that this can present like this, or you can have this edge as straight, but this edge could be thicker here like this. Okay. And this was also irregular acanthosis. So what we used to do is, if you can put a straight line touching the downward growth of the epidermis. If you can just put a straight line, for example, here it is irregular acanthosis, because if you put a straight line, it is not a straight. It doesn't go straight. The thickness varies. For example, here it is thinner, but here it is thicker, the same as here. So this is irregular acanthosis. Okay. I have photos available where you see regular acanthosis, so we'll discuss that again on those photographs. For example, here, this is the involved slide, and you can actually draw a line like this. And this is regular acanthosis. Okay. So some people might say that why it is regular acanthosis when this area is thinner. This area is not thinner. It is actually normal. It is the same as this. It is the same as this. It is the same as this. But the dermal papillae, not the dermal papillae, but the epidermal downward papillae are reaching to similar heights. This level is nearly same. So it is regular acanthosis. Okay. The entity which I wanted to show in this photograph is known as hypergranulosis. Okay. Hypergranulosis. If the granular layer is more than five layer thick, and I'm talking only about the granular layer, the blue granular layer. If it is more than 5 layer thick, it is hypergranulosis or increased granularity. And here you can see that the granular layer is very thick. So if you zoom out, you can actually see that this is layer 1, this is layer 2, this is 3, this is 4, this is 5, and this is 6. So you have a 6 layers of granularity, and that is what is hypergranulosis. Let's move forward. The second entity is hypogranulosis. And here in this picture, you will see that you cannot find the granular layer. Okay. What you can see is compact hyperkeratosis, isn't it? You can see the compact layering of the cell, but the cell is, look at this. Look at this stratum corneum. Look at the stratum corneum. It is thicker than one third of the epidermis. We are clear about it. This is the entire epidermis, and it is thicker than one third of epidermis. So hyperkeratosis is there. Clear? Not all layers of the epidermis is thickened. So acanthosis is not there. But you cannot, you can see one layer here, one cell here of granular layer, but you cannot see any other layer, any other cells in the granular layer. There is no stratum granulosum. And that is why this slide is showing hypogranulosis. You will see it in disorders like lichen planus, but this picture is more of ichthyosis, where you have retention compact hyperkeratosis with no granularity or no granular layer. Okay, so now you understand the absence of granular layer is also very important. It is usually about 3 to 5 layer thick. More than 5 layers, you should think of hypergranulosis. The next pattern is spongiosis. So can you see this empty space here? Let me see if I can use another pen. Not visible. Not visible. Okay, look at this here, this area here. Can you see this empty space? This is a vacuole, and it is seen in spongiotic tissue reaction pattern. So in spongiosis, the appearance of the epidermis is like a sponge, because what exactly is a sponge? You have a cuboidal space and you have multiple air spaces in between, isn't it? That is what a sponge is. And that is how an epidermis looks like when there are multiple empty spaces. So here is one empty space, here is one empty space, here is one empty space, here is one empty space. And it is there in the level of stratum spinosum. Okay, this is the granular layer. So stratum spinosum has a lot of empty space. For example, look at this area of the slide. Now this area is normal, but here you have a spongiotic focus, okay? And again, you have the normal area. So can you now appreciate the spongiosis? I'll just get rid of these markings so that you can actually see the spongiotic foci. Okay, so now can you appreciate the spongiotic foci? And that is spongiosis. It is seen in disorders where the inflammation is happening in the epidermis, this vesicle formation, this oozing, like in dermatitis. That is the pathognomic of or the prototype of spongiotic tissue reaction pattern. Dermatitis and inflammation of the skin. Let's move forward. Now, many a times in the last slide, we have seen that these empty spaces are actually empty. Well, they are not. They are filled with fluid and fibrin and serum and all sorts of tissues, but the cells are not there. The empty areas are devoid of cells. But what happens when these are populated or invaded by different cells? Not exactly invaded, but you find different cell population in these empty spaces. For example, in this photomicrograph, what you're looking at are neutrophils. These are newts. So if you look closely, let me see if I can zoom it out. Yeah. So if you look closely, you will see the cells have multilobulated nucleus. The nucleus are there in multiple lobules. Okay. If you look closely and carefully, you will see that these cells are not keratinocytes. They are smaller than keratinocytes and they have multiple lobules in the nucleus, making it a more multilobulated cell. And that is why, because they are neutrophils. And when neutrophils are there in the spongiosis or the spongiotic vesicle, we call it as neutrophilic spongiosis. Okay, let's move forward. Similarly, in this slide, you can point out, you can see that in the spinous layer, let me change, yeah. In the spinous layer, you can see this empty spaces in between, empty space here, empty space here, empty space, because it is spongiosis. But like neutrophils, you can have eosinophils. So can you see this cell is a little bit red? This is a little bit red. This is a little bit reddish color as compared to surrounding area. This is red. This is red. Why? Because now they are not neutrophils. They are eosinophils. Okay. And eosinophils have a large unlobulated nucleus and they have reddish eosinophilic cytoplasm. Okay. On the other hand, neutrophils are larger and they have a basophilic multilobulated nucleus. Basophilic multilobulated nucleus. So this is a newt, this is an eosinophil. And here you can see the spongiotic vesicles have eosinophils. Let's move forward. The next reaction pattern is papillomatosis. In papillomatosis, what actually happens is you have elongation of the downward projections of the epidermis. Okay, so normally this area, the portion of the dermis which is going up, is known as the dermal papilla. Dermal papillae. The downward projection of the epidermis is actually epidermal papilla, or we just say it as epidermal projections. Okay, epidermal papilla. What happens in papillomatosis is that you will have elongation of this epidermal growth and the uppermost layer will be undulating, going up and down and up and down and up and down. Okay, up and down, up and down. So it goes in a wave form. Okay, it is actually like this. And this going in a wave form is known as papillomatosis. Okay, here you can see a dermal papilla destruction. Dermal papilla microabscess is here, spongiosis here. Maybe this is a slide of, I don't know, dermatitis herpetiformis, but that is why you see papillomatosis. In subcorneal split, see the uppermost layer or the roof of the split is of stratum corneum. There are no nucleus in the cell, sorry, no nucleus in the layer, while all the other layers of the epidermis, that means the granular layer, this is the granular layer, the spinosum layer, and the basal layer is below the split. So the split has happened. If this is the stratum corneum, this is the granular layer, this is the spinous layer, and this is the basal layer. So the split has happened below the stratum corneum, and that is what is known as subcorneal split. You can in fact see one neutrophil here in between the split. And this is how a subcorneal split will look like. The roof will be because of the roof will be made of stratum corneum, while below it will be all the other layers of the epidermis. This is an intraepidermal split. So here you can see you have stratum corneum, you will have the granular layer, you will have a little bit of spinous layer and a little bit of spinous layer below, and the basal layer. So this is the basal layer, spinous, granulosum, and corneum. So here the split will be in between the layers of the stratum spinosum. So here you can see that in this diagram, the split is in between the spinous layer. And that is known as intraepidermal split. Okay. We will not be going into which disorders show these features because right now the motive of this video lecture is to sensitize residents on how to look at a diseased and a normal epidermis and try to find the findings first. When you collect all the findings, then you make a decision on what exactly disorders you are looking at. So let's move forward. And here we have a supra-basal split that we see in pemphigus. Okay. And in supra-basal split, again, if we make the same diagram, you have stratum corneum, you have granular layer, you have the spinous layer, and below the split, you have the basal layer. So here is the split. Okay. So below the split, you have the basal layer. And in pemphigus, you see the row of tombstone appearance. So if I make it, it's like this. Like in a cemetery, you have tombstones. So they look like tombstones in a row because they have lost their attachment to the surrounding cells because of destruction of desmoglins. And that is what you're seeing. You're also seeing a little bit of fibrin exudates here. You are also seeing neutrophils in, sorry, acantholytic cells. These rounded cells are acantholytic cells. We'll discuss them when in subsequent future we'll be discussing pemphigus biopsy. So acantholytic cells, and this is how a supra-basal split will look like. Okay, it will look like stratum corneum, granulosum, spinosum, and then below the split you will have stratum basale. Okay, let's move forward. This is an intraepidermal pustule. It is like a large spongiotic vesicle, very large pustule. And why are we calling it as pustule? Because it is filled with newts, filled with neutrophils. So it's a neutrophilic intraepidermal pustule. Mostly it is seen in pompholics. If you biopsy a pompholics, you will see this kind of a localized collection filled with neutrophils and large empty area filled with neutrophils. Let's move forward. So we have covered the major epidermal reaction patterns, how epidermis looks in different pathologies, and very quickly we'll go to minor epidermal reaction patterns. These are those reaction patterns which you don't encounter frequently, and they're quite pathognomic of different disorders. We will not be discussing the disorders, we'll just go through how they look like under a microscope. The first is dyskeratosis. Now, as the name suggests, dyskeratosis means abnormal keratinization. This means abnormal, and keratosis means keratinization. That means the keratinocyte, because of one reason or the other, has not been able to properly perform the function of keratinocytes or keratinization. The cell has not been able to properly keratinize, and because of improper keratinization, the cells have changed their morphology. So these are known as dyskeratotic cells. If the dyskeratotic cells are present inside viable epidermis, that means the epidermis is normal, but in between the epidermis, you have these dyskeratotic cells. They are known as corporons, which means round-bodied. Okay, corporons. These are larger cells with irregular eccentric nucleus surrounded by a clear halo with brightly eosinophilic cytoplasm. Okay, so here if we see, it's a large cell. It has an eccentric nucleus. Okay, you have a clear halo around the nucleus, a clear halo like this, and you have eosinophilic cytoplasm, bright eosinophilic cytoplasm, and this is how a dyskeratotic cell will look. Dyskeratotic cell. Okay, and they're usually present in the granular layer. Remember, when the cell reaches the granular layer, it is rich in the basophilic granules. The organelles are begin to disintegrate. You won't see them if the process is normal. If the process is not normal, you will see accumulation of altered keratin filaments, and the cell is not able to properly keratinize, and that is what leads to dyskeratotic cells. Okay. If the same dyskeratotic cells are found in stratum corneum, okay, so in stratum corneum, they're known as grains. Because why grains? Because now they're smaller and they are flatter. Okay, so if let's say this is the cut section, you have the basal layer, you have the spine, you have the spinous layer, you have the granular layer, and then you have a flattened cornocyte. If the problem occurs at the level of stratum granulosum, you have corporons. If it occurs at the level of flattened stratum corneum, you have grains, like grains of rice. They are more eosinophilic, flatter structures. Focus on the flatter structures with scanty cytoplasm because they have reached the stratum corneum and abundant keratohyalin granules. So that is the difference between coprons and grains. And dyskeratosis is mostly seen in disorders like Darier's, Galli-Galli, Grover's disease, Warty dyskeratoma, Haley disease. So we'll discuss how it looks in under a microscope, but we will not discuss individual disorders. So here we can see in dyskeratosis, what actually do we look at? We look at the intraepidermal split. So here is a split. Why a split has occurred? Because the cells are not properly formed, and that is why the layers separate from each other. If the process would have been normal, for example, here the process is normally occurring. There is no problem here. So you see the normal biopsy, but here you can see the cells are not normal. You can see one dyskeratotic keratinocyte. You can see another dyskeratotic keratinocyte. And because the cells are dyskeratotic, you are actually splitting the epidermis because they are not closely glued to each other. The cells are not normal. And that is why you are looking at dyskeratosis. Okay, and that is why the split and dyskeratosis are combined and together they are known as acantholytic Dyskeratosis. So inside the split, you can see one acantholytic cells, another acantholytic cells, here also acantholytic cells. So these are all acantholytic cells. In fact, here you can see a column of parakeratosis. Okay, so that makes us realize that it's a scaly disorder which has erosions because of abnormal keratinization. Let's move forward. So here is a magnified image of acantholytic dyskeratosis so that you can see the coprons here. So this is a copron, this large cell. You have a thickened nucleoli, thickened nucleus, a halo around it, and the cell which it doesn't look normal. Look at this. In the prior slides, we have discussed the normal appearance of epidermal layers, but here the cells do not look normal. They look out of place. And they are present at the level of stratum spinosum and stratum granulosum, isn't it? So these are known as coprons. Let me just write it here again. Coprons. Can you see how round they are, round bodied? That is why coprons. While the flattened one are the grains. So here also you can see the dyskeratotic cells. Here's a dyskeratotic cell, dyskeratotic. These are all dyskeratotic cells. They are rounded cells. Remember, at the level of stratum spinosum, you will have shrunken cells which are forming spinous process. At the level of stratum granulosum, you have flattened spindle-shaped cells which are more basophilic granules. And because of that, the appearance of large round cells is very conspicuous. You can easily find them out. So if you have a split in the epidermis in the biopsy, look at the split and see if you can find out acantholytic dyskeratosis. In the meanwhile, in the comment section, tell me what is this cell here. Here. This cell here. See and let me see how many of the viewers can actually recognize this particular cell. Just mention that in the comment section. Cornoid lamella. This is the second minor reaction pattern. Now remember, it is cornoid. Cornoid lamella. It is not coronoid. There is only two, there are only two O's in it. Okay. No third O. There is no coronoid. Okay. This is wrong. It is cornoid lamella. Cornoid lamella is a thin column of parakeratotic cells with an absent or decreased underlying granular zone. Okay, so what actually happens is this is the normal stratum corneum. Let's say it is basket weave normally. And then you have an invagination, not exactly invagination, but a column, a pillar. Columns and pillars are different architecturally, but just for the understanding, you will have a column of parakeratotic nucleated cells. Okay, parakeratotic nucleated cells. And below the parakeratotic nucleated cells, the granular layer would be absent. So this purple is stratum granulosum, and just below, where it is just about the tip of the coronoid lamella, the granular layer is either absent, it is not there, or it is just deficient. Okay, so that is how you describe a coronoid lamella. It is because there is a localized area of faulty keratinization. That means the keratinocytes at this particular area, they have no idea how to keratinize properly, and because of wrongful keratinization, it has led to formation of a parakeratotic pillar. Am I making sense? Is it easy to understand now? And clinically, this presents as a thin thread-like raised border, which we see in porokeratosis. So in porokeratosis, you have this kind of a border thread-like. And why thread-like? Because a column of parakeratotic cells are there. The skin is not normal there. Okay, let's move forward. So in this picture, let me just change the color. Yeah. In this picture, you can easily appreciate this column of parakeratosis. Okay, you can see the nucleus inside the stratum corneum. You can in fact see a little bit of granular layer outside. Granular layer just adjacent to the column. You can see granular layer here, but there is no granular layer at the bottom of the pillar. So that is cornoid lamella. In fact, in the second picture, you can see this is the parakeratotic column, and here is the granular layer. Can you see this granular layer here? And it is also present here, but it is broken off. It breaks off just below the column of parakeratosis. Let's move forward. Epidermolytic hyperkeratosis. Now, in epidermolytic hyperkeratosis, you have compact hyperkeratosis with granular and vacuolar degeneration. Okay, that means the cells inside the epidermis, they have a lot of empty spaces, and the stratum corneum above is actually thickened with compact hyperkeratosis. Let me show compact hyperkeratosis like this. So you have thickened compact hyperkeratosis along with granular and vacuolar degeneration of the cells of the spinous and granular layer. Okay, and inside this split, you will see reticulate lightly staining amphophilic material with distinct cellular boundaries. What do we mean by that? We mean that if this place, let me just erase it and draw it again. We have compact hyperkeratosis, and in between the epidermis, you have a large split. And inside the split, you will have amorphous, pinkish, lightly staining, some kind of pale pinkish appearance of substance in epidermolytic hyperkeratosis. You can also have dyskeratotic cells. So here you can see. Let me just change the color. Yeah, you can you see here this kind of spongiosis like appearance, and you can easily see the compact hyperkeratosis, the thickening of the stratum corneum, more than the 1/3 of the epidermis, isn't it? So you can appreciate a bit about epidermolytic hyperkeratosis. Now, epidermolytic hyperkeratosis in a varied different disorders. It can be localized, generalized, severe. All those clinical disorders can present with epidermolytic hyperkeratosis. It can in fact be an incidental finding also. We'll discuss epidermolytic hyperkeratosis in future videos where it plays a much more important part. So with that, the lecture is more or less finished. I would just recommend these two books if you are just starting dermatopathology. It's a very, very good idea to read Livers and Weedons. Okay, you can read Livers and Weedons. I think they are as important as Rooks and Bologna as far as dermatopathologists are concerned. We also have Mackey's. That is also a very good book. If you can get access to it, do read it. The two bare minimum books that I would recommend is IDVIL textbook of dermatopathology and Fundamentals of Pathology of Skin. Okay, these two books are very good. They're available on Amazon. There is an ulterior motive to recommend this because I have a chapter on spongiotic dermatitis in this book. Uh, so just go through these two books if you really are interested in dermatopathology. Actually, you should go it. This is the bare minimum for dermatopathology. If you're really interested, you should read these books. Okay. So I hope we have made a little bit of sense what exactly is there in a normal epidermis under microscope, and what exactly are the different disorders of the epidermis and how they look like in an H&E stained slide. Reading recommendations, of course, Weedon's, Lever's, Fundamentals of Pathology of Skin is very important. These two I would really recommend if residents can go through this, and then subsequently go to reference books. This tadpole article is good. This small article about epidermal reaction patterns are good. But if you have gone through this video, you will have a very decent and strong understanding of what different layers look like and what different pathologies of different layers look like. So without much further ado, we have just gone through the first of the series in dermatopathology. I don't know if the series will be continued or not, depending on how much response this video actually gets. It's a long video. It's nearly an hour long, and the most important aspect of understanding dermatopathology is go and look at the slides. Okay, every teacher says that go and look at the slides, and every student thinks it's useless. No, you have to go and look at the slides. You will not understand if you look in on the internet, because on the internet you will see textbook prototypic slides. You need to know how normal slides look, and you need to get adjusted to the act of actually viewing the slides in a microscope using both of your eyes, using different magnifications, using different foci of slides, or the level of slides, or how to set a stage, how to actually use the oil immersion. Know everything about it, and you will gradually understand all of this pinkish and bluish hues will slowly start to make a little bit of sense, and then you just keep on looking at slides and keep on learning new and new things. So without much further ado, we'll finish this lecture. Adios and bye bye. We'll see you next week with some other topic. Bye.